Good afternoon. My name is Gary Kettner. I'm a professor in the W. Harry Feinstein Department of Molecular Microbiology and Immunology and a virologist, and I'm going to sort of be moderating the session this afternoon. So let me be the first to welcome you to what is a dual celebration, really, of the contributions of basic biological sciences, and in particular microbiology, to public health. As you probably all know, when the school was founded in 1916, along with the departments which dealt with topics which were relevant to public health and known to be relevant to public health, such as epidemiology, William Henry Welch, the founding dean, established a series of departments which dealt in basic sciences, because he appreciated the fact that most public health problems are underlain by biological problems, that the understanding of the biological problems was essential for an effective public health response. And so, in addition to, for example, epidemiology, he established a Department of Immunology. I'll come back to that. Bacteriology, within a few years, he had established departments which evolved into parasitology and disease ecology and virology. So since the founding of the school, those biological basic sciences have continued from strength to strength, and it's the accomplishments and the achievements of those departments and the school as a whole that we're here to celebrate today. So there'll be two parts to the celebration. The second one, which will be this afternoon at 4 o'clock, will be the recognition by the American Society of Microbiology, which is the professional society for microbiologists, of the school as a whole as a milestone site in microbiology for contributions made in my department, MMI, and in other departments, including epidemiology and biochemistry. That ceremony will involve an address by Dr. Ellen McKenzie and also by the president of the ASM. And as I say, that's going to be at 4 o'clock. For now, what we're going to do is concentrate on two subdisciplines, if you will, of the ongoing work in the school. Virology and immunology, and we're going to concentrate on two firsts, and those two firsts are the first Department of Immunology, which, as I said, was established at the establishment of the school in 1916, and the first Department of Virology, which was established later in 1925. So as a virologist, my heart is, of course, with the Department of Virology. The first course in virology, not just here, but anywhere, was taught in 1922 by a virologist named Charles Simon. And the success of that course among students at the school led, as I say, to the establishment of the Department of Virology in 1925. As I say, we'll follow these two threads, immunology and virology, for the remainder of the session here. So the welcoming remarks will be made by Dr. Diane Griffin, former chair of molecular microbiology and immunology, currently a university distinguished professor and vice president of the National Academy of Sciences. Dr. Griffin came to Hopkins as a virology fellow to the Department of all places, neurology, to pursue an interest of hers in the departments in the study of viruses which infect nerves. And she established, along with her colleagues there at that time, a subset of virology, which we now refer to as neurovirology. As I said, Dr. Griffin was chair of MMI for almost 20 years, and she actually is the person who planted the seed for this celebration with the notion that we really should remember and memorialize the 100 plus years of virology at the School of Public Health. So, Dr. Griffin.
All right, well, it's a great pleasure to be the welcoming person for this celebration. And as Dr. Kettner has mentioned, this has been in the works really since almost before the pandemic, as we realized that we were at 100 years, and that was a mark that should be celebrated. And as Dr. Kettner has also emphasized, the founding vision for the School of Public Health in 1916 from William Welch, who was both a bacteriologist and an immunologist, was that fundamental research to actually understand the biologic basis of disease was necessary in order to be able to eventually come up with interventions. Being able to actually control those diseases required a basic understanding of how the diseases happened and how they happen. So those founding principles have been basically carried on throughout the last 100+ years. So the very first departments, there were 7 of them in the School of Public Health. And as Gary has mentioned, in addition to the sort of more classic School of Public Health types of disciplines, there were 4 basic science departments, bacteriology and immunology. And as I mentioned, Welch was both a bacteriologist and an immunologist. So he named himself chair of both of those departments. And then there was medical zoology, which was basically parasitology, but also vectors, mosquito-borne diseases, and understanding those vectors. And then chemical hygiene, which of course was really biochemistry. And those were all established at the very beginning. But it was followed soon thereafter by the Department of Filterable Viruses, which was based on the first course that was taught about viruses. And then that evolved basically into the Department of Virology. Now, one of the consequences of this sort of special relationship that the basic sciences had with the School of Public Health was that, and I don't know enough about this background, and maybe we'll get more information today. The university did not allow the School of Public Health to give a PhD. You could study any of these disciplines, but the degree that you got was an SCD rather than a PhD. So you'll see that Dr. Roisman, our keynote speaker, is an SCD. Harry Feinstein, who established an endowment for the department subsequently, and the chair of the department also had an SCD. So I'm not sure when that transition to being able to give a PhD occurred. So these founding principles for the School of Public Health they've fostered a wide variety of studies, and I'm just listed a few here, and on the opening slide there were many more. But it really has engendered the ability to apply basic science research to public health problems. And we're all proud of the accomplishments that have occurred. So next I'm going to introduce Arturo Casadoval, who is the current chair of the Department of Molecular Microbiology and Immunology, who will also give welcoming remarks and introduce our keynote speaker.
Thank you, Diane, and thank you for your leadership in this department for so many years. It is my pleasure to introduce our keynote speaker, Dr. Bernard Roisman. He's one of the most important virologists of the 20th century, having made contributions to the biology of many viruses. His name is on the plaque that will be unveiled today as part of the group that discovered respiratory syncytial virus. I have known Dr. Roisman for about 20 years. I first met him and had the opportunity to talk to him when he was in the scientific advisory board of the Northeastern Biodefense Center that I was the co-PI with Ian Lipkin. And at dinner, I remember that he told me his story of how he had come to the United States, World War II, and all the things that had happened. I remember saying to him, you got to write this down. Now, I don't know if that had an effect or not, but he did write it down. and I had sent it to the faculty. And in 2015, he published this remarkable story of his life, and it includes, for us here at Hopkins, a very detailed description of what it was like, this department was like in the mid-1950s. He has had a stellar career. He's our alumnus. He's a member of the National Academy of Science and has received innumerable awards and honors and awards. So I'm not going to rattle them, but instead, I thought I would tell you two anecdotes from the article that I think give you a sense of the remarkable trajectory and the person. So here he's describing his visit to Hopkins. My trip to Hopkins was entirely different. Before, he was talking about his trip to Yale and his trip to Harvard. Microbiology was in the School of Hygiene and Public Health. I came to Baltimore by train and to the department office by noon. Tom Ward, soon to be my mentor, informed me that he played bridge every day at noon and that he could not see me before 1 p.m. He also informed me that if I joined the lab, I would have to learn to play bridge because one of the players was about to leave. The interview was short and to the point. When I asked Tom Ward what the project he had in mind, to me, his response was that I could do whatever I wanted. I accepted on the spot. Learning bridge seemed like a small price to pay for the opportunity to work on viruses. So he came to the department in 1955. He did his PhD rapidly. Dr. Ward left him in the department. From what I can tell, he appears to have supervised himself. There is a great description in his article of his PhD defense in which apparently nobody read his thesis. And after 1 hour when he was waiting, he was very worried, but instead the department chair went outside and offered him a job. So he went straight from being a graduate student to being a faculty member. But before he could get his PhD, he had to complete courses. And it was found that he had not had all his courses. And I will read you another small paragraph from the book. At the end of the year, I was one course short before I could graduate. The only course available to me dealt with the rat populations in Baltimore. My task was to estimate the rat population within a block close to the center of the city. I set traps, marked the rats I caught, and released them. This was repeated several times. In the end, we estimated the block housed roughly 300 rats. Other blocks in the city fare worse. So at lunch today, we asked him, how you actually get the number of rats to be 300. And he explained that what you do is you catch a rat and you mark it and you release it and you keep catching rats. And after a while, from the number of returning rats that have been marked, one can estimate the rat population. With that, I think I've given you an essence of a remarkable Dr. Roisman, and I will turn it over to him, who's going to tell us about some of the work that he did here. Thank you.
I'm very pleased to have this opportunity to come back to the school of my dreams. I want to tell you that for me, this institution has been cornerstone to my career as a scientist. I learned a lot here. And and just to give you a little bit more of a background, what happened is in 1954, 70 years ago, I applied to Yale, to Harvard, and to Hopkins. And the two institutions, Harvard and Yale, I somehow didn't find them attractive. They were very formal, very difficult to communicate with. My interview was very pleasant, but nevertheless, I didn't accept them. Hopkins was a totally different story. I was told to wait while Tom Ward finished his game of bridge. He explained to me that that he played bridge whenever he was in town at lunch, and it was very stimulating. I promised that I would learn how to play. I'm a chess player, and bridge just didn't fit. But it wasn't necessary. Shortly after I came, Tom Ward began to be absent from the lab for long periods of time. And every time I talked to him about my project, he kept telling me, later, my lad, don't rush me. And so the end result was that I never really spoke to him about my project. And less than a year after I came, he left, which left me with a problem. I could accept another advisor, or I could pretend that I don't need one, and that's what I did. Formerly, Tom Hubner, the chairman of the department, acted as my advisor, but he was not a virologist. I went to see him about once a month for about 5 minutes. That was it. My work was primarily involved a lot of biostatistics, and I had a lot of help from them. Now, there's one other item that I should tell you about. I had to defend my dissertation. It was, that's to say, 2 days before I took the prelims. The next day I took the language exams. There were 2 languages I had to show efficiency. And next I had to defend my dissertation before Dissertation Advisory Committee at that time. I was very nervous. I somehow managed to insult one of the examiners who didn't understand the dissertation, and the committee asked me to leave, and I'm done. So I went back to my lab, convinced that I was finished. Imagine that after 2 hours later, Tom Hubner came to my office and asked me if I had a job. I told him I didn't have one yet, but I'm sure I could get one as a technician. I was convinced I failed. And he said, no, you didn't fail, you passed. I have a job for you. Go to work with Manfred Mayer, an immunologist, on the polio project. We just got the grant from National Polio Foundation, and this is an opportunity for you to become part of this organization. So let me tell you what happened. First of all, let me tell you about the problem. At that particular time, there were sporadic epidemics of poliomyelitis in the United States, particularly in densely populated populations. And they can cause a lot of damage. For example, the 1908 epidemic in New York City killed 9,000 people. 2,400 people. they were mostly children, and there are thousands more with lifelong disabilities. And I should point out that many parents at that time sent their children out to the countryside to avoid exposure to polio. The solution, obviously, was a vaccine. And the initial vaccine was the counter vaccine. And you might remember that the result was a disaster. The result was 40,000 cases of myelitis and 230 children paralyzed and 4 dead. This is out of 100,000 vaccinated people. Obviously, this was not a good vaccine. Something better had to be done. And there is another thing I should tell you. There were two principal characters in the field at that time, Jonas Salk and Manfred Mayer. Most of you probably never heard of Jonas Salk or Manfred Mayer. Albert Sabin. But they were real characters, very dominating, very difficult to convince that they were wrong. And so I began to work in the lab, and I had to learn a few things. Manfred Mayer was an extraordinarily good quantitative immunologist. His strength was in complement fixation. He brought it to an art, and it was used to quantify proteins, antigens. If there was an antibody and you had the proteins, you could purify, or a mixture, you could purify the protein of choice. And so we began to construct two things. We began to construct antibodies for using complement fixation against polio, and develop techniques for measuring the results. So the next slide shows how we have done this. There was the title of it is an identification of antigen-inducing protective immunity. And the way it was done is to infect the tissue culture cells with extract of infected tissue. They were then harvested and homogenized the infected tissue culture cells. We would then layer the homogenate on top of a sucrose density gradients and centrifuge to equilibrium. And I should tell you that at that time, Manfred Mayer's lab was one of the few that had analytical centrifuges. We had to develop skills to use them. They at that time were not commonplace, and they were the only centrifuges at the university at that time. So what we would do is to layer the homogenates on top of the sucrose density gradient and centrifuge to equilibrium. And the gradients containing extracts of infected cell generally show two prominent bands, which designated C band and D band. So I want to tell you what came out of this study, and I switched. What came out of this study is that characterization of C and D antigens yielded very interesting results. The C antigens were retained in the infected cells. They were not excreted. They were not type specific. Type 1 C antigens reacted with type 3 or 2 antibody. The D antigens were exported from the cell. they were infectious and they were type specific. So we have two sets of reagents. And it was obvious that if we were to monitor the amount of antigen we give on vaccination, it was very important to have the antigens quantified and used properly. There was a lot more. We were interested in how these virus antigens function. And we didn't get very far. We could tell that the antibody that was produced was effective. Polio literally disappeared from the United States, and we had a powerful vaccine on our hands. But for me, it raised very important questions. Namely, what was the role of CD, C antigen? Was this C antigen the useless product? What was left over after the D was formed? Was this cDNA the stepping stone to D, or the remaining protein from after D was made? And the data generally suggests very strongly that the C antigen is a leftover, is part of a release particle, a particle that releases DNA or say RNA and left behind the shell, modified with respect to specific activity. And my interest also comes from another point of view. I have been working with herpes viruses, and one of the interesting things is that herpes virus incorporates its DNA into capsids at the nuclear pore. So the empty capsid is outside the nucleus, the DNA is inside, properly exactly the opposite of what we expected, and it's then sandwiched into the particle as it comes along. And simultaneously, there appears to be an immunologic change. And we, to some extent, began to understand what is going on. Where is this leading me to? It will be very helpful to have a way by which we put therapeutic viruses into the body and have them stay there until appropriate moment and then induce the release of DNA. For the past 10 years, my laboratory has been interested in producing viruses for therapy, for the destruction of cancer cells, for introducing new genes into cells, and so on. And it would be nice to synchronize the whole thing using a way to block the release of DNA until the right moment. We were beginning to understand this, but we are still far away from, I shouldn't say far, we're a little bit away yet from using them directly in people. I'm going to stop here and try to answer questions.
Thank you very much, Dr. Reisman. The contributions that Hopkins made to the development of the poliovirus vaccine are going to be dealt with in more detail this afternoon in a talk by Marie Hardwick, I believe. Okay. So Kirti Shah fresh from earning an MBBS degree in India, met Frederick Bang, who was to be chairman of MMI later in India while Bang was there doing field research. Dr. Bang was apparently a persuasive individual, and he convinced Dr. Shah to come back to Hopkins. Kirti obtained both his MPH and his DRPH degrees from Hopkins. And after being sent off for seasoning at the Rockefeller and then briefly back to India, was invited back to Hopkins as a faculty member. Dr. Shah was a quintessential virologist. He had a broad interest. He worked on arboviruses. He worked on small DNA tumor viruses. But he's really best known for his work on human papillomavirus, in which he was able to contribute some of the evidence that made it clear that human papillomavirus was the etiologic agent of cervical cancer. Dr. Patty Gravitt was a postdoc or as a graduate student in Dr. Shah's lab whilst getting her PhD in epidemiology, and was there during the heyday of the development of assays for HPV that eventually made it possible to unequivocally determine that HPV was the etiologic agent of cervical cancer. And so Dr. Gravitt, who is now at the NIH, Deputy Director of the Center for Global Health, will speak about her recollections of those times in Dr. Shah's laboratory and perhaps even other things. Thanks.
So thank you, Dr. Kettner, and I had no idea I was going to be following such a preeminent scientist, so I'm a little put off by this. But really, I am so honored to be invited here. I couldn't think of somebody I more want to honor than Kirti Shah. He was such a fixture, I think, in MMI for so many years. And I just want to share some of the things that I have. I didn't write anything scripted, but to just honor him in a few different ways. So fortunately, I don't have to say too much about this. Gary's already told you that Kirti got his MBBS in Pune, India at the BJ Medical College, and then subsequently his MPH and PhD at the Johns Hopkins DRPH at the Johns Hopkins University. And from 1962 until he retired in 2013 was faculty MMI at the Johns Hopkins School of Public Health, and all of MMI's and Johns Hopkins name changes. One of the things that I found when I was kind of looking for a few publications and things on the internet is I came across a Google site that said, "What does the name Kirti mean?" And I thought, well, that's kind of interesting. What does Kirti mean? Never thought of it. So here's what Kirti means. It's fame, a famous person, reputation, happiness. And I thought, I don't know if Kirti's parents knew what he was going to become, but how incredibly appropriate were those meanings to the man that I grew to know and love dearly, almost as a father. So I'm going to go through some of what makes Kirti Shah famous, what we all know him for, but then I'm really going to talk a lot more about what Kirti's reputation was to all of us who worked with him and learned from him, and then kind of hopefully tell you kind of what I believe his happiness was, and it comes right down to all the work he did. I'm going to start off by saying Kirti was a prolific scientist. One of the things that I always say to the people that I mentor, because I don't know how many of you guys are, on an APT committee or have suffered through APT committee, but the people care about certain metrics. They care about how many publications you have. They care about how much citations you've had. Kirti published over 400 papers in his career. His H-index was 112. H10 index was 367. You can look at the citations per year. Kirti was a prolific scientist, but as I was being mentored by him, not one time, not a single time, did Kirti ever sit down and advise me, you've got to get this paper published. You've got to get this in a high impact journal. You have to do these things. Kierty talked to me about the science. Is this interesting? Have we done a good job? Do we need to do another experiment? And then just get it done. The biggest thing that he did for me, though, is to advise me, Patty, you talk too much and you write too little. So that was very good advice. And sadly, if Kierty is listening now, I'm no better at that than I was before. But I want to say this because we can all be, kind of chasing these metrics, but a really good scientist who's just seriously curious will succeed. And he did, without chasing the metrics, without chasing the fame and the glory, and yet he achieved it. His humility makes us all not understand how much he did, but he did quite a lot. And obviously what he's best known for is the causal relationship between human papillomavirus and cervical cancer. So I started in my master's degree in molecular biology at University of North Carolina at Charlotte in 1988. And that's when we were really just starting to understand that papillomaviruses were probably or possibly a cause of cervical cancer. Kierty worked with a team from the IARC, it was an international team, and really did fundamentally use the molecular biology techniques at the time. Anybody want to guess what those were in the early days? Anybody remember doing Southern blots? So using Southern blots really to isolate and understand and see that we can extract papillomaviruses, not just from the cervical cancer tumors, but he's also the one who isolated HPV-11 from papillomas and really nailed down a very rare but debilitating disease, recurrent respiratory papillomatosis, as being caused by certain strains of HPV infection. He was a basic science leader in that international team that confirmed the causal relationship between HPV and cervical cancer. And, that work, which you can see was published in 1999, 2002, has gone in the last 2 decades to lead the WHO to have the first call for the elimination of a cancer as a public health problem. This is something that's phenomenal. I mean, thinking as a virologist that you're actually going to be able to eliminate a cancer from the face of the earth, I mean, who would have thought it, right? And it was really Kierty's work that fundamentally at that time helped. And I spoke with Nubia Munoz right after Kierty passed away. And she said, Patty, we sat there and we fought and we argued. And at one point, Kierty, just in his very, very, easygoing style, said, enough. The evidence is there. The evidence is there. This is the cause. And she said at that point, they all said, let's write this paper. Human papillomavirus is a necessary cause of invasive cervical cancer worldwide. This was largely, and you can see Kierty's not the senior author, he's not the first author, because that's not what was important to him, but he was a key part. And that really shows too, Kierty's amazing ability to take the basic science and the virology and translate it so elegantly to the epidemiologist. And he kept them on their toes. He kept them on their toes to make sure that they understood what they were looking at. They understood what they were measuring and how to infer something about it. It was an amazing time. He then went on. I can remember coming here in 1997, I think, right before I moved here. I was interviewing at University of Washington and Hopkins for my PhD in epidemiology. And so I came and met with Kierty, and he said, oh, Patty, you should look at papillomaviruses in oral cancer. And I said, no, there's so much more to do in the cervix. I really want to do that. And he's like, okay, well, I made a very bad mistake then, I think. The next thing that Kierty did after he helped to establish the causal relationship between HPV and cervical cancer is he worked with a colleague, Mara Gillison, to really establish that papillomavirus infection, particularly 16 and 18, were a fundamentally important cause of oropharyngeal cancers as well. Again, one of the stories that I was told after Kirti passed away, and I was writing an article for HPV World, which is our Papillomavirus Society newsletter. And I spoke with Mara, and she said, I'm going to tell you a story I don't think anybody knows. And apparently she and Kirti were trying to get funding to support their work on looking at HPV and cervical cancer. And she said, we were running out of money. And Kirti stopped taking some of his salary so that he could support her and he could support the work to keep this going until they could secure funding. And this has been an amazing paradigm shift in how we think about HPV-associated cancers, how we think about oral cancers, how we treat oral cancers. Kirti opened his lab to Mara and I. We had faculty members who, at least in epidemiology, having a lab scientist was something that was very new and nobody knew what to do with me. Kirti opened his lab to me. He opened his lab to Mara when the School of Medicine didn't have a lot of space. And we worked together and it was just amazing. And Kirti, again, kind of just stood behind the scenes and supported two women scientists to really excel. And Mara has gone on to do great things. And this was another big kind of fame-inducing set of work that Kirti did. The next thing is something that actually comes a little bit ironically from the poliovirus. So one of the early poliovirus lots were actually contaminated with simian virus 40, SV40. And then in kind of, I guess, the early days of medical misinformation, there was a lot of theory that brain tumors and lymphomas were actually being caused by people who were vaccinated with these lots of contaminated SV40 vaccine. Kirti stuck his neck out more than once, and I had been in conversations with the very serious discussions that he had, but really sat down and did the most difficult thing that we can do in science, which is to prove a negative, to prove that there was really no evidence to suggest that this contaminated vaccine caused cancer. This is another thing that we don't really recognize Kirti's work on, but was really something that he was very passionate about. And I'm going to end with talking about his reputation and happiness. When I started reading the obituaries when Kirti passed away, Diane, I think, wrote a lot. I think Noel Rose wrote some things in response. And everyone said the same thing. Kirti was a quintessential mentor. And as somebody who had the great honor to be mentored by him, he really was. Kirti was transdisciplinary and interdisciplinary before I think we knew that was a term. He really understood how to link fields together in ways that were amazing. I think he probably mentored more epidemiology students across his tenure in the last few years than he did MMI students. He was just a delight to work for, to work with. He was so supportive. He always stayed in the background. But that does not mean that he was easy. He was very hard. He was hard on me. He would challenge me at every step, and he would let me fall. I don't think this is going to work, Patty, but go for it. And, when I fell, he would be there to say, yeah, so what did we learn? Kirti was truly an amazing mentor. I couldn't say how much I miss him as a result of that. He didn't want to take glory in anything. And, until the day he died, he was, texting Alvaro Munoz or emailing him, asking him, how do we design the study to show that human papillomavirus vaccination will prevent recurrent respiratory papillomatosis? It's a rare outcome. How can we prove that? He always cared, and he was genuinely happy and excited to do the work that he did. If I came in with a, random new little thing, he would be so excited and want to hear all about it. He, and I can't talk about Kirti and his lab without mentioning Rick Daniels, who also passed away in the last few years, and Joy Mays. And Rick and Kirti, I think, were innovators before their time. I mean, I've heard that Rick, fashioned a PCR machine out of water baths back in the early days. So really innovating in that way. I know that when I first came, I came from Roche Molecular Systems. We had just started developing TACMAN technology at the time. And we brought, the first, I think, real-time thermal cycler to Kirti's lab. So he really was on top of the new technologies, knew where they would be useful, where they could be applied. And, it's the genuine curiosity and delight in doing science that honestly, I don't see in many people anymore. It's too much of a rat race. It's too much of competition. I have to always go back. Kirti gave me a Buddha head. It's a bronze Buddha head. And he came, when he was leaving for Florida, he was giving away a lot of things in their house. And he said, Patty, come. And I said, what? And he said, I want you to have this. Farida does not want me to give it away. And Farida was his lovely wife. And I treasure that more than anything, because every time that I look at that, I think of that peacefulness, genuine curiosity, love of life, and happiness that Kirti had. And in terms of happiness, there's not anything in the world that made me happier than I would come into his office, and almost every single time there would be a call. Oh, sorry, that's Farida. Pick up the phone. Hello, sweetie. And, just someone who cared that much about the students that he had, the passion that he gave to his work. It's no wonder that he is as famous as he is, that he was as successful as he was. And I think the message I especially want to give to the younger students in MMI now, you can do that with sheer passion and curiosity. You don't have to be so strategic. You can succeed. It doesn't maybe happen as fast, but everything that he instilled in me, I hope that I continue to move forward to this day. Thank you, Gary and Diane and Marie, for inviting me here. I really appreciate it. And I do miss Kirti terribly, but he gave us all so much. Thanks.
Thank you, Patty. My lab was immediately next to Kirti's when I first came here, and he did, in fact, have a PCR machine connected to water baths. And I remember Rick Daniels sitting in front of this thing for hours, connecting the reaction tube by means of manually operated valves, first to a hot water bath, and then to a reaction temperature water bath, and back to a denaturing water bath for hours, sitting in front of this machine with valves. Kirti was interim chair of the department twice, which I think sets a record. A man who inspired enormous affection and respect. Noel Rose came to MMI as chair in 1982. Noel was the first individual to show in an experimental animal system that immune reactions to an individual's own proteins were capable of inducing disease. And what he did was to induce a disease we now call autoimmune thyroiditis in mice by injection with thyroglobulin. He's known for this reason as the man who really founded autoimmune disease research. And he continued to conduct experiments in autoimmunity for his entire career. He, as rumor has it, he held a continuously funded NIH grant for 60 years. And it is, I think, emblematic of both of his right sense of humor and his consideration for the rest of us by saying, again, allegedly, that he finally decided not to renew that grant in order to give the young people a chance. Daniela Chirkova came to Dr. Rose's lab as a postdoctoral fellow. She's now a professor in pathology, and Dr. Chirkova is going to share reminiscences of Noel's lab with us now.
Thank you very much for the invitation and the opportunity to give a tribute to my mentor, Dr. Noel Rose. So Noel started his scientific journey in Dr. Vitebsky's lab. And at that time, it was considered preposterous to think that there could be something like an autoimmune disease. So autoimmune disease is when your own body attacks your own tissue and causes a damage and long life disease. And these diseases mostly affect women. In fact, in many of these diseases, predominantly women were affected by the autoimmune diseases. So at that time, the main people in the immunology field, like Dr. Paul Ehrlich, who received a Nobel Prize in medicine for basically setting up the basis of immunology, they're not believing that something like autoimmunity even can exist. So Nell Rose, so I don't know why it got bigger, but now we can focus on now a little bit more. So he is the handsome, dark, tall man with Vitebsky, his mentor, on the side. And you can see the drawing of the thyroid gland. So thyroid is right here. And they chose, thyroid unknowingly. That actually what a good choice it was. The autoimmune thyroid disease is the main autoimmunity that people get diagnosed with. And they, two of them, set up to a journey to show that there is indeed a thing as autoimmune disease. And Noel spent years to really prove it to very skeptical immunology establishment at the time that could really happen. He used rabbits. He showed that you can take the thyroid gland from a rabbit and immunize another rabbit with it and cause inflammation. And Vitebsky was very skeptical and said, you really have to show that it's the autoreaction, so against really the self-tissue. It's not good enough from one rabbit to another. So he made him to do a microsurgery and actually cut half of the thyroid gland, make an immunization out of that to the same rabbit, and then show that this rabbit had an autoimmunity in the remaining part of the thyroid gland. So it shows you the rigor of the science that Noel was doing to really persuade everybody that this is indeed happening and there is such a thing as autoimmune diseases. So now we know that there is more than 80 autoimmune diseases, and this is really thanks to Noel and his mentor Vitebsky. So Noel was often called father of autoimmunity, and I think really deservingly. But Noel also, because rigor and scientific accuracy was what he really was standing for always. He also really was ticked off by a phenomenon that was happening at the time they discovered autoimmunity, and it's still sometimes happening. So when there was a new disease discovered that nobody knew the origin of, it was often called, it's maybe autoimmune. So he didn't like that because he said there needs to be a strict scientific criteria that we will be using to define what is autoimmune disease and what is not. So he went on and set up such a criteria. They are known as Vitebsky-Rose autoimmune criteria. They are listed there. And basically, just to summarize them, he wanted to see that the autoimmune disease could be either transferred from a human to human, for example, by transfusion, or from mother to a child, or could be replicated in an animal, in a model, or could be transferred from sera to an animal. And he also had indirect kind of evidence that this is an autoimmune disease. So he always stood there, to make sure, yes, we name all the autoimmune diseases, but he also defined them very well. And we are very sure about identifying what is autoimmune disease and what is autoimmune disease is not. So I have some numbers for you. So as you heard, that rumor is indeed true. That he was 60 years uninterruptedly funded by NIH until he retired in 2015 in his 88 years of age. He was also chair of Autoimmune Disease Coordinating Committee in NIH. And when I joined the lab as a postdoctoral fellow, at that time, the internet was not as accessible as now. And I went to his office and I asked can I please get just a list of his articles so I can start reading before I start the job? And I was given something like that. And I said, no, I don't need the articles, just the list. And the secretary said, this is the list. So 880 articles. He was such a prolific writer, and he has amazing memory. To his 90s, he would dictate articles. Like, I cannot imagine how he did that. I couldn't certainly do that. But he remembered all the citation from memory, from 1950s to 2000, right? And it was any time able to actually pull it out from his memory. So this is how he wrote articles. He just dictate them to the dictaphone and have secretary to write them when he was, in his 80s, 90s, because he just remembered all of them. He was editor and co-editor of 22 books, and he was very proud of his autoimmune diseases. It was a huge book that he wrote with many co-authors and co-edited with Dr. Jan McKay. And I just listed some of his awards. There was no room for all of them. We would be here for another half an hour, so this is just a pick. And as it was mentioned, he was a chair of the Department of Immunology here in School of Public Health for 10 years. But I also listed his other appointments because he was also a full appointment in the Department of Pathology, so in both School of Public Health and School of Medicine. And that just shows, how active he was. And he really wanted, to do things and do them everywhere. And he was, he started a division of immunology and was chairing it in pathology. And that division still exists. And it's a basic research in pathology with 7 faculty that I am part of. He was also very active in the Senate, presided in the School of Medicine. He directed Center of Autoimmune Disease Research and organized wonderful autoimmune days from 2001 until he retired that were really well attended by everybody that meant something in autoimmunity. He always said that his research was really divided between the heart and thyroid. So first 30 years, he mostly dedicated his work to thyroid disease, and the last 30 years to the heart. And I found this picture from 2007 of his lab. He's there in the center, and on his right side is Dr. Katuregli, who is a professor in pathology, and he exemplifies the thyroid part of the focus of Noel of his research. On his left side is Dr. Burek, that together with Monica Talor, that is standing just behind Noel, were directing a clinical lab that he also started. So besides being a prolific researcher, Noel was also really big in clinical laboratory medicine. And he also started criteria and ways that clinical lab should be governed. And he also started first board for clinical laboratory directors. So he, really was in many ways, involved in immunology, not only in the research, but also in the clinical laboratory immunology. And then also there is me in standing in the back when, I started with him working on the cardiac autoimmunity, and we went together to do many articles and many projects. And I want to just highlight 2 main kind of areas in the heart disease that we did together. So on the right side is eosinophilic myocarditis. So this is autoimmune disease caused by eosinophils that is very destructive to the heart and can happen to the people that are otherwise healthy, or can happen to people that have allergies, and they will have lots of fibrosis, and they could also develop a heart failure. So you can see the box is the articles that we published, in this area of research. And on the left is the second area I want to mention. We discover a new pathway that leads to heart failure, and it's driven by a mediator called IL-17 that can damage the heart and cause lots of fibrosis. So you can see there are 2 hearts. One of them is very blue. That's fibrosis. That's damage caused by the cytokine IL-17. And when you don't have the cytokine in the IL-17 knockout mice, the heart is protected and doesn't develop fibrosis. So these are the 2 main areas of the heart that we work on together. Noel was always mentoring, and this is one of my favorite pictures of him. This is in his office in Rose Building. And he's like very gleaming at that picture and smiling because the student was coming. And that was, I think, a favorite part of his day, to talk to the students, what they are doing, in the lab, and just, chat about the daily activities in the lab. So I found this quote in many of the obituaries that went in press after he died. From Dr. Mackenzie, former dean of Johns Hopkins Bloomberg School of Public Health. She said, "Noel was an extraordinary scientist, an exceptionally kind and humble human being, always looking out for the well-being of faculty and students." And this is an old picture of him teaching. And a quote from him, he said, "The main lesson I learned is that teachers are there to serve the students, not to demonstrate their own accomplishments." And I thought about it and thought how to finish, my tribute today to him. And I wanted to finish it with a legacy and with the students. So he started the autoimmune heart disease research. And it didn't die with him. It didn't stop with his retirement. These are all students from MMI that graduated recently, and all working on autoimmunity. And many of them, as you can see from the pictures below, that are their main papers, working on autoimmune heart disease on the projects that basically came from him. So you see Nicola Dini, that is her last graduate student that I commented with him, and her publication in eosinophilic myocarditis. Snow Hu and her publication of interplay of macrophages and fibroblasts, Megan Wood on arthritis, Hanna Kalinowski on tissue-resident T cells in the heart. Abdel Hamad is a master's student that graduated in this year, working on a 3D model of the very model that Noel started. And Won Yang Yu on another new model of autoimmune myocarditis, this time driven by cancer therapy. And I want to end with this 3D model of the mouse model of autoimmune myocarditis that Noel developed 30 years ago. And the master student this year from my lab designed to the 3D of the micromillimeter, of the micromolar model like that. And I think Noel would really like it. You can see in the purple is all the inflammation, all the autoimmune inflammation in the heart. The red are the vessels. The gray are cardiomyocytes. And I hope somehow he can see it and is smiling down at us and wishing us luck continuing in working in autoimmune diseases. So thank you so much.
So virology and immunology are threads which follow MMI and its predecessors through our history. They aren't, however, the only things that go on in MMI. And so we're going to have now 7 short talks from individuals in the department who do things other than immunology and other than virology. The talks are short, and I'm going to ask each of the people to come down and introduce themselves, except for the first individual, Nicole Baumgarth, who's a newcomer to Hopkins and who has a conflict and can't be here, but who has recorded a video. Dr. Baumgarth is a DVM and a PhD and a Bloomberg Distinguished Professor and director of the Lyme and Tick-Borne Diseases Research and Education Institute, which is a descendant in a way, I guess, of medical entomology that was one of the departments established at the founding of the school. So Dr. Baumgarth will be here by video, and I believe the next speaker is going to be Dr. Carlton.
Hello, my name is Nicole Baumgart. I'm the inaugural director of the Lyme and Tick-Borne Diseases Research and Education Institute. This is a new institute that is embedded within this department. It is the brainchild of Karen Peets and David Nolan, 2 Johns Hopkins trustees who were personally affected by tick-borne diseases and who approached Arturo Casadeval. Arturo saw the need and for such an institute, since we know that ticks and tick-borne diseases are on the rise. even though not much has been researched about it. Nearly 95% of all vector-borne diseases, that is, diseases carried by small critters, in this country are caused actually by ticks, not by mosquitoes. Yet very little is known about them. Very little is known about how climate change will impact them. And so this institute is established at a fantastic time. And I was recruited from California to take on the challenge to establish this place. We're very excited to develop this institute within the Bloomberg School of Public Health, because at the end of the day, what we're trying to achieve is bend this curve that is currently going up with increasing numbers of tick-borne diseases every year. Currently, about 500,000 affected people every year, and most of them affected by Lyme disease. Now, I'm sure most of you know somebody who's affected by Lyme disease, but there are other diseases that are carried by ticks that we understand much less, Powassan virus and others. We need to know more about them. We need to know what the risk is for acquiring them. This is why our institute has a mission to provide basic research to work on the ticks, the pathogens they carry, and how the diseases are occurring, but also to provide an accurate assessment of the risk from acquiring a tick-borne disease, and then eventually develop mitigation strategies so that we can find better ways to prevent tick-borne infections, which there are currently no vaccines available. So with this vision, it includes work in my own lab, where we are trying to understand antibody responses, B-cell responses, trying to develop ways to improve on the humoral immunity that occurs after vaccination. And we are also studying in mice Borrelia burgdorferi infections, that is infections with the Lyme disease agent, to try and understand how these bacteria are evading immunity, how they're hiding and suppressing immune responses to establish persistence, with the eventual goal to develop therapies that can work in conjunction with or instead of antibiotics to clear and Lyme disease or the pathogen from hosts. With that, I would like to finish by thanking Marie for all the hard work she put in getting this event organized, to dig through all the history books, to show the rich history and impact this department has. And we hope that the ITBDI will contribute and add to this history. Thank you very much.
Great. My name is Jane Carlton. I'm the new director of the Johns Hopkins Malaria Research Institute here at the Bloomberg School of Public Health. And we are an alliance of scientists in a state-of-the-art facility doing high-impact research on the malaria triangle, the mosquito, the parasite, and the human host. Our goal is to innovate the strategies that prevent and cure malaria. I should say that the Institute was founded in 2001 by a generous gift from Bloomberg Philanthropies. Our key assets, and I'm hoping this video is going to work. Let's see. Yes, so our key assets include our mosquito insectaries and malaria parasite core. As you can see here, these mosquitoes have been fed on blood, which enable the female mosquito to lay eggs, and subsequently those hatch into larvae, and then they subsequently hatch into mosquitoes. And it's the availability of the thousands of mosquitoes each week that really enables our researchers to undertake sophisticated and controlled experiments, for example, to see how the parasite develops in the mosquito and how it transmits the parasite. We have a designated set of staff, the mosquito makers, as we call them, and our mosquitoes are world famous and provided to other labs for acquiring them. Because the mosquito is so important in the transmission of the malaria parasite, we have a big focus on developing methods for controlling mosquitoes. So, for example, George Demopoulos, shown here, is genetically modifying mosquitoes to prevent them from transmitting malaria. Marcelo Jacobs Larina is infecting mosquitoes with natural bacteria that secrete molecules that are toxic to the parasite. And then also Conor McMenamum, who you're going to hear about a bit later, is identifying human scent chemicals and how the mosquito detects them. We are also developing novel vaccines and therapeutics. A group of investigators led by Terry Shapiro recently repurposed a known drug and developed it as what's called a chemical vaccine. Matt Ippolito is testing new drugs in combination with the frontline drug used in Zambia. And Drs. Fidel Zavala and Prakash Srinivasan are generating antibodies and vaccines that protect against malaria. The Malaria Institute serves the public and the scientific community with conferences, monthly podcasts, and YouTube videos. On the left here, you can see a flyer for a recent World Malaria Day symposium that was held just last week, Omics on the Move. And this is held every year on World Malaria Day, which is April 25th. And here you can see Vector Encounter, which is happening next month, where groups of vector biologists get together just with a whiteboard, no presentations, no fancy PowerPoint is allowed for discussions. And then we have later in the fall, the Future of Malaria Research, where younger malariologists come together to describe their research. And I should say Malaria Institute scientists don't work in a silo. We are not stuck in our ivory towers. We partner with researchers at African and Asian field sites to combat malaria. In particular, we have a very large footprint at the Matcha Research Trust in Zambia with an insectary and a 5,000 square foot mosquito sphere and molecular biology lab. And myself and Dr. Bill Moss are directors of 2 NIH International Centers of Excellence for Malaria Research in Zambia and India, where we're developing tools and strategies to decrease malaria. And if this works, which I hope it does, so this is a video of one of our field sites in India, in the state of Odisha. This is in a clinic. You can see the clinician here is taking a blood sample from a person infected with the malaria parasite. And this is now being processed in the lab and the DNA extracted. And we have brought over new technology, a portable sequencer, as you can see there, that a PhD student is then loading that sample onto. And you'll see in a short minute the DNA A, G, T, and C coming off so that we can identify the genetic diversity and the evolution of the malaria parasite. So I hope this has given you a sort of snapshot, really, of the research that we are undertaking at the Institute. Together through cutting-edge research, teaching, and training, we are training the next generation of researchers. We are committed to leading science for a malaria-free world. Thank you.
Thank you. Fungi are fascinating organisms capable of thriving in extreme environments, from polar deserts to radioactive sites like Chernobyl, and even outer space. Some fungal species have been found growing inside spacecraft and surviving more than 1 year and a half out in the outside of the International Space Station. And it is this resilience to radiation that inspired us to begin exploring the potential use of fungi in protecting against radiation in space, which is a big problem. Since 2019, we started collaborating with NASA, and here is a picture of our first fungal biomaterials being tested outside the International Space Station. Where humans go, fungi follow. And with increasing interest in human space travel, understanding their uses and potential threats in space is crucial. This emerging field of study is known as astromycology. Fungi's coolness isn't just figurative. This is a study that we published last year where we use an infrared thermal camera to measure the temperature of more than 40 different fungal species. In the infrared images you see, warm areas appear in yellow and red, while cooler areas show in dark blues. All the species that we measured, that we imaged, appear colder than their surroundings. Fungi cool themselves via water evaporation, similar to human sweating. And mushrooms are so good at this, at evaporative cooling, that it inspired us to invent an air cooling device we call the mycocooler, which was recently patented, by the way. I think that studying how fungi regulate temperature could potentially help us figure out how to control their growth and perhaps also develop novel antifungal therapies. Now, here's the twist. While fungi typically remain cool, they appear to warm up when facing bacteria. In this experiment, we infected fungal mycelium with bacteria and observed a temperature increase that was proportional to the bacterial concentration. Although it is preliminary, we're excited because this hints at a fever-like mechanism in fungi. And if confirmed, it would suggest that fungi possesses an immune system and it could serve as models to study fever and perhaps enabling us to develop novel therapies. So fungi are not just cool, they're cosmic adventurers, temperature regulators, and perhaps even fever fighters. Thank you.
I'm just switching to the laser pointer. Great. Thanks so much for the opportunity to speak today. I'm really excited to give you a feel for what we've been doing in the lab. So in my lab, we're interested in how bacterial heterogeneity, which is driven by immune cell interactions, also impacts antibiotic treatment efficacy. And we've been using 2 different bacterial pathogens in the lab to study these interactions. And so what I wanted to show you here is our model with Staph aureus. We've been focusing on methicillin-resistant strains and using a mouse model of kidney abscess formation to visualize interactions between immune cells and different bacteria that are present. So here is a kidney and the arrows, here. The arrows are pointing out the bacterial communities that are replicating within this tissue. If you zoom in to a single lesion level, you can see the bacteria are surrounded by different immune cell subsets, including macrophages. And we can also utilize fluorescence approaches in the lab to visualize changes within the bacterial population. What's been really interesting about this is that we can see that interactions with specific immune cells drive changes in the bacterial population that I'll tell you a little bit more about on the next slide. Our Yersinia model is shown here on the bottom, where we again work with a mouse model of infection. This is a natural pathogen of rodents, but also causes human infections as well. We can visualize the bacteria replicating within tissue sites. We can visualize interactions with different immune cell subsets. We can detect different phenotypic changes in the immune cells as well. An example of that is nitric oxide production here. And we can utilize fluorescent-based approaches to understand how the bacteria are changing as well. So this becomes really important. We should have something on the bottom, yeah. So this becomes really important when we start to think about treating these infections and administering antibiotics. On the left side, I'm again representing our Yersinia model, where we know that the bacteria are changing in response to neutrophils and in response to monocytes. Again, one of the pathways that we've been very interested in is the response to nitric oxide. This is a response specifically to recruited monocytes, and what we find is that when we treat with antibiotics such as doxycycline, the cells that are stressed are surviving at higher rates. And we found that this response to nitric oxide is one example of how the bacteria are predisposed to survive. But what's been very interesting in this model system is that we can initially reduce the bacterial load and it stays repressed for a certain period of time. But then when antibiotic concentrations wane, the bacterial population is capable of rebounding and causing a relapsing infection. And so we're very interested in how the bacteria are surviving in the presence of the antibiotic and also some of the mechanisms the bacteria are utilizing to cause this rebound, with the idea that maybe we could identify some pathways that we could target simultaneously with a second antibiotic. In our other model system with staph, one of the aspects of it that I wanted to highlight was that Staph aureus is specifically a human pathogen, and so we've been setting up different models in the lab to be able to understand human cell interactions with the organism. And so in this model, we're incubating bacteria with human neutrophils and allowing the bacteria to set up these types of abscess structures, as I showed you in the mouse model, where the bacteria are clustered. What's very interesting is when you try to treat these abscesses, initially the bacteria are very sensitive to the antibiotic, but as these structures fully form, the bacteria become very resistant to the impact of the antibiotic. And what's important here is that these strains are already methicillin resistant. What we're finding is that they're also exhibiting vancomycin resistance, and that's what's typically used to treat these infections in the clinic. And so this model is also going to be very powerful moving forward and allowing us to identify the different mechanisms that are at play here that allow the bacteria to survive this antibiotic treatment. And then just to wrap up, I wanted to acknowledge some of our current lab members that have done the work that I showed you today, and some of our previous and current lab members that generated some of the images I showed you on the first slide, in addition to the data on the second slide, and to highlight some of our external collaborators and funding sources. Thank you.
Okay, hi everyone, I'm Connor McMenamin, and research in the McMenamin group focuses on understanding the sensory biology of mosquito attraction to humans. And so pictured here, feeding on my finger actually, is the most deadly animal in the world, the African malaria mosquito, Anopheles gambiae. And in the McMenamin group, we're really fascinated to understand the molecular and cellular mechanisms that this mosquito uses to find humans to transmit malaria parasites. Indeed, the transmission of malaria is exclusively driven by the lust of this particular female mosquito species and its intimate association with skin and the associated scent profile. So in our group, we use integrative methodology to try and understand the chemosensory biology of this process. To locate humans in the environment, mosquitoes first smell you, and then they subsequently fly along plumes of body odor that we emit. Getting into proximity of humans around 5 to 10 meters away, they can see you. And then with about 1 meter, they can actually sense the warmth from your skin. Over the past few years in our group, we've developed innovative methodologies to profile what we like to call the whole body volatilome, all of the volatile or airborne chemicals that are emitted by the human body. And so seen here on the left of the screen is just a heat map of a cohort of 20 individual humans, 10 men and 10 women, where we've profiled some of the most abundant chemicals emitted by the human scent signature. We're currently exploiting this method in northern Zambia to understand the impact of Plasmodium falciparum infection, the causative agent of cerebral malaria, on the human scent signature. And we're finding some very interesting things, including that certain chemicals in the scent signature are altered by the presence of infection. In our lab, we also couple this with state-of-the-art genome engineering methods to try and understand how the olfactory sensory neurons in the mosquito olfactory system are tuned to these components of the human volatilome. Potentially by understanding this information, we can help to develop new methods to combat malaria in a very low-cost way. And so towards that, we've been developing synthetic blends of chemicals that smell like very attractive humans. Pictured here is a very low-cost adhesive trap where we've engineered a blend of chemicals that smells just like your average human. In the future, we hope to advance upon this to sort of yield the next generation of very targeted approaches to trap malaria mosquitoes and potentially other anthropophilic disease vectors. Those are the ones that are the most dangerous for human health, to combat both malaria and other vector-borne diseases globally. So thanks.
Okay, thank you. All right, thank you. My name is Sean Prigge, and I'm really pleased to be part of the celebration today. And I also feel privileged to have known both of the investigators when I was a young faculty member who are honored today. And I'm also very privileged to be part of this historical recognition from the ASM. I'm actually starting to feel a little historical myself because I realize I've been here 20 years. So in the 20 years that my lab has been here, we've been focused on studying malaria parasites and in particular the metabolism in these parasites. And very quickly this work started to focus on nutritional requirements. And so we became interested in what nutrients do the parasites need to take from the human host in order to survive. And so I've been studying these nutrients, and when we find ones that are essential for parasite survival, our next question is, well, what is it so important that they're doing with these nutrients? And so that leads us into essential processes that the parasites have to do in order to survive. And of course, the goal for this sort of work is to ultimately come up with ways to either block the parasites from getting these nutrients or how they use them. And so fortunately, we have a lot of tools to do this. So it's shown here on the, here we go. What's shown here on the right are parasite cultures. So we can grow these parasites in blood stage cultures in the lab. And this allows us to do genetics, biochemistry, we can measure metabolites, we can measure the gene expression, these parasites, and a variety of techniques using this model. And so I'm going to talk about today in 2 slides are work that we've done and really the take-home message for these 2 metabolites, one that you may not be familiar with, lipoate, and another one that a lot of you may take in your multivitamins, which is vitamin B5. And these metabolites have different roles within the parasite cell. And so I've shown this simplistic view of the parasite to highlight the fact that there's multiple compartments in each cell. There's a nucleus, mitochondrion, and another compartment or organelle called an apicoplast, which actually has an interesting connection to plant chloroplasts, which you wouldn't expect for a pathogen. And all 3 of these are relevant to the biology of these 2 metabolites. So in terms of what we've concluded from these metabolites, looking at the first one, lipoate is a molecule that's made by humans. This is a human metabolite that we make. And we discovered that the parasites require this in order to survive inside of our cells. And they use it exclusively in their mitochondria. And so we tracked the biology, the important biology here to the mitochondrion. And we found that the essential thing that they're doing in the mitochondrion is making this metabolite called acetyl-CoA. But it's not essential for the mitochondria in the parasites. It's actually used elsewhere in the cell. And the aspect of it, or the use of it, that we're most excited about and really is driving future work is the role that acetyl-CoA plays in the nucleus to drive parasite gene expression. And so the take-home message from this project is really that a human metabolite is used by the parasites to control their gene expression. And then the second metabolite that I'm going to talk about is pantothenate, vitamin B5. And so this, of course, people don't make. It's a human vitamin. And so the humans get this primarily through their diet, and the parasites then require getting this from the humans in order to survive. And this metabolite is ultimately used exclusively in the apicoplast organelle. And here it's used to synthesize that molecule I mentioned in the last slide, coenzyme A, or CoA. And in this case, this project has led us to a very practical direction, which is working with collaborators to screen for inhibitors of the enzyme that we discovered catalyzes this process in the apicoplast. And it also led us to a more broader appreciation for the parasites. This is in another project, actually, where there appears to be a connection between human nutrition, the vitamins that people are taking in their dietary source, and the ability of the parasites to survive in the human host. Thank you.
All right, I'm the last one. Hi, everyone. I'm Yotam Suez, and our team studies the human microbiome in health and disease. And if I have a pointer, we can also skip between slides. Today, I want to share with you a vignette from a project that covers a lot of topics that we're interested in the lab, including the interaction between the microbiome and diet in the context of cardiometabolic health, and also the implications for personalized medicine. So in this project, we're asking how non-caloric sweeteners like aspartame, stevia, saccharin, sucralose impact our cardiometabolic health. We know that these sweeteners don't contain any calories and they don't directly impact blood glucose levels, which makes them attractive alternatives to sugar in people that are trying to lose weight or maintain healthy blood glucose levels. And we definitely can see here in this chart that the number of products containing artificial sweeteners, the number of people consuming these products is constantly on the rise. And at the same time, also the number of people with obesity, counterintuitively, is also on the rise, which led to the hypothesis that actually sweeteners might contribute to the same conditions that they aim to treat or prevent. However, evidence in humans for a causal link between sweeteners consumption and an impact on cardiometabolic health are very heterogeneous, and we were interested in identifying the cause of that heterogeneity. So in a clinical trial that we published in 2022, we saw that the impact of sweeteners on both blood glucose as well as the microbiome was personalized, meaning that some people responded worse than others. We were able to link the extent of the impact of sweeteners on the microbiome to how worse was the impact on blood glucose levels. And this really brings up a lot of interesting questions for us. So first one is, how do sweeteners are able to change the microbiome? And why are some microbiomes responding to sweeteners while others are not? The sweeteners are not able to change these microbiomes. And then another question that we're interested in is, once the microbiome has been altered by artificial sweeteners, how is that new microbiome, this dysbiotic microbiome, impacting our cardiometabolic health? So to answer these questions, we cultured a complex microbiome community in a test tube and we exposed it to artificial sweeteners. And using metagenomic sequencing, we were able to demonstrate that artificial sweeteners can directly alter the microbiome. And potentially they're altering the microbiome because they can inhibit the growth of that complex microbial community in a dose-dependent manner. But it's really interesting to note that not all bacteria respond the same. So when we culture gut bacteria, when we produce monocultures of gut bacteria and we expose them to sweeteners, we can see that some bacteria are very strongly inhibited, but others not so much. So this potentially explains why some microbiomes respond and others not. If you have more of these bacteria, maybe you're more likely to experience an impact of sweeteners on your microbiome as comparison to someone that has more of these bacteria here. Oh, I'm the only one who's seen my pointer. Anyway, another interesting observation is that once we take that microbial community that's been altered by sweeteners and we transplant that community into germ-free mice, we see that this alone can lead to exacerbated weight gain and disruption of blood glucose, which is mirrored in both the fasting blood glucose levels and also a glucose tolerance test. So in order to understand what's going on here, we collected a lot of data from these germ-free mice, a lot of omics data from these mice. And what we can see is that having these two distinct microbial communities that were altered by sweeteners can then lead to the accumulation of various immune cells in the liver, which is an important metabolic tissue. And this leads to various differences in gene expression in the liver and also differences in metabolites that we can find in the gut of these mice. And now we're trying to see how all of these differences in those various omics can explain the phenotype that we're seeing, the metabolic phenotype that we're seeing. Thank you.
All of which makes MMI an extraordinarily interesting place to be. So there's one last speaker before we break, before the plaque presentation, and that's Reggie Jackson, who's a member of the Office of External Affairs, which he characterizes as a unit integral to alumni relations and development work for the school and the Department of MMI, and which in fact has contributed in some cases to the research you've seen presented here. So, Reggie?
Thank you, Gary. It's so interesting to hear from so many talented researchers and faculty about everything from putting down your vitamin B5s and diet sodas to space fungi. So quite interesting. But it's a pleasure to see so many of you here today to celebrate this special moment. We have over 1,100 alumni living in over 30 countries. And that makes each of you a part of this lasting legacy. So thank you. You know, in our department, there are many ways that we engage, from fundraising and also to alumni engagement. So we welcome you to share your stories, where you are in your careers, your research, and to connect you with each other, other students, other faculty, where you live, or even remotely. So the BSPH alumni team, in collaboration with the department, is committed to helping, committed to helping strengthen the public health network. And we would welcome your thoughts on how to make that happen. I'm simply Reggie J at JHU, so you can reach out to me or any one of my team members that you may meet. And finally, I hope that you continue to enjoy the festivities today as we move toward a quick break for laboratory tours, as well as the plaque unveiling and the reception later on today. But please feel free to connect with me again, or anyone within the Office of External Affairs, and we would love to find ways to creatively connect you with each other. Thanks.
All right, so we'll take a break now, and we'll reconvene in 15 minutes, 4 o'clock, for the presentation of the plaque that honors or recognizes the entire School of Public Health as a milestone site in microbiology. So we'll see you in 15 minutes. Okay, if you'll be seated, we're going to go ahead and get started. So please have a seat. So as I mentioned earlier, the pursuit of biological science in service of public health is a school-wide pursuit, and so the designation of the School of Public Health as a microbiology milestone site is an award for the school and not simply for our department. And so the dean of the Bloomberg School of Public Health, Dr. Ellen McKenzie, the 11th dean, it turns out, is going to speak briefly in introduction. Right.
Well, thanks, Gary. And it's so wonderful to see everybody here on such a wonderful occasion. It's just great to have the opportunity to celebrate yet another milestone moment for the Johns Hopkins Bloomberg School of Public Health. We're so proud to be recognized as an historic site by the American Society for Microbiology, and we're honored to have President Virginia Miller with us here today. Dr. Miller, thank you for all you do, and especially for lifting up the history of science, which is so important. So thank you so much for that. I also want to thank our esteemed alumnus, Bernard Roisman, for being here with us today. I had the distinct pleasure of having lunch with Dr. Roisman at his home in Chicago. I was trying to think of when it was, but it was a few years ago, quite a few years ago. We did not get into a lot of deep discussion about virology or microbiology, thank goodness, because I am not an expert in those areas. But we did talk more broadly about some of the challenges in public health. But what I remember most, Bernard, was listening to your story and your journey through life and your recollections of your extensive travels and looking at all the wonderful things you had in your home that you picked up along the way in your travels. It was such a wonderful, wonderful afternoon, and I remember it with great fondness. So thank you so very much for being here. It really, really means a lot to the faculty, to the students, to the school. Thank you for being here. I also want to thank the speakers from earlier this afternoon who presented and gave us a little bit of history of the school and some of the wonderful things we've done along the way. You all provided us with a powerful reminder that immunology and virology have protected health and saved lives on an extraordinary scale, shaping modern life in the process. And of course, we're here to celebrate the foundational role that our school played in that progress. So allow me to take you a little bit back in time and remind you that our school was founded back in 1916 with a gift from the Rockefeller Foundation as the world's first independent graduate school of public health. Soon after, we established departments of immunology and epidemiology, and again, these were global firsts. During that very first year of the school's existence, if you recall, 1916, influenza had begun to sweep across the world. By the time the first classes were held in 1918, the flu had become one of the deadliest pandemics in human history, killing tens of millions. While the Bloomberg School was then in its very earliest days, its faculty led research and response efforts, both locally, but also nationally and internationally. As just one example, Wade Hampton Frost, who was our founding chair of the Department of Epidemiology, devised a method that provided the first accurate picture of the pandemic's mortality toll in the US. And amazingly, the core of his methods are still used today, more than a century later. By responding to the 1918 pandemic, the school jump-started its growth as a leading center of research and training in epidemic disease. We were led through that challenging and formative time by our first dean, William Henry Welch, a prominent bacteriologist and pathologist. He famously said that all of our social problems, all of our industrial problems, all of our economic problems are all related to problems of health. That truth has stood the test of time, and we have seen the Bloomberg School evolve to truly embrace it. We continue to explore the complex connections between public health and other disciplines and sectors of our society, and by doing so, address some of the biggest, most formidable challenges that face humanity. At the Bloomberg School, our work extends from laboratories right here in Baltimore to maternity wards in Bangladesh and to the malaria field stations in Zambia and way beyond. We now have more than 900 full-time faculty members. At any given time, we're training more than 3,000 students from around 90 countries around the world. Our 28,000 alumni live around the world, and they multiply our impact by working on the biggest, most important frontiers in public health. Our faculty are advancing life-saving gun policies, reimagining our food systems, and bringing mental health interventions to communities in need around the world. And of course, they are continuing to lead the way on game-changing discoveries in microbiology, immunology, and epidemiology. Time and again, the Bloomberg School has made history, and we continue to make history with every passing year. To be recognized by the ASM for these milestones is really a true honor. It is a reminder of our shared mission to harness the power of science for the good of humanity. I want to thank you all for being here to celebrate with us. And we look forward to, I guess the plaque has already been unveiled, but we'll unveil it again. But thanks again for being with us here today. And now it is my tremendous pleasure to welcome Arturo Casadevall, our esteemed chair of molecular microbiology and immunology. Arturo?
Thank you, Ellen, for those wonderful remarks. When I came to the school in 2015, I happened to be very interested in history, and I began to realize that there was a lot of history here. In particular, contributions to the fields of microbiology, infectious diseases, and immunology, and that had come from the School of Public Health, and particularly the departments of epidemiology and MMI, which dated to the beginning of the foundation of the school. So then, when I was traveling, I went to many places and I saw plaques. I don't remember exactly where was the place that it inspired me, but I thought we needed to have a plaque to celebrate things. And the question you could ask is, why? And what is the value of history? And I will put to you the value of history in an anecdote from the early 20th century. So those of you who remember history, prior to World War I, there was a naval race between England and Germany. England was the master of the seas, having established supremacy at sea in the Battle of Trafalgar in 1905 when it defeated the combined fleets of Spain and France. In the 1890s, the Germans were mounting a major challenge, building a brand new fleet. And the English, the British, reassured themselves with the following saying that applies to what the departments. You can build a fleet in 10 years, but it takes a century to build a navy. What do they mean? It means that you could build the ships, but you can't build a tradition without history. Therefore, for example, think about departments today. If you have enough money, you can get yourself a building, you can get yourself hire people, and you can put together a great department doing great science. But you cannot buy the history for that department. History binds us together. History gives us a sense of identity. And therefore, I thought it was really important to try to do this. And the question was how? Well, I was very lucky in that my colleague, Marie Hardwick. Marie, many of you who know her, is a tremendous scientist, and we began to have discussions, how to do this. And in particular, what to celebrate, because think about it, a lot happened. What do you put in? What do you leave out? When do you set the date that you're going to celebrate up to? And Marie began a process, and this process took many years. She, in the way that those of you who know Marie, the first thing Marie did is she read the instructions and to figure out what was needed. And those of you who know me, you know that that's the last thing I do. So we kind of complement each other very well. She put a committee. The committee included Peter Agri, Carrie Debbing, Diane Griffin, Gary Kettner, Al Scott, Dave Sullivan, and myself. And we began to meet. And we began to talk about what were the things to celebrate and what were the findings. And it turns out that, and this is something that I want to address tonight, it is very hard often to reconstruct history because scientists don't preserve their history. But for example, when we were looking at the discovery of RSV, I challenge you to go to the papers and try to figure out exactly what happened. No, papers, as Peter Medawar wrote, are often a lie. They don't represent the process of discovery. So working with Karen Thomas, who's a historian, and after a lot of discussion, an application was put together, and that application led to today. And I want to acknowledge that this would not have happened without Marie, and that we owe her a tremendous, basically gratitude, because she's given both the departments of epidemiology, molecular microbiology, and the school a tremendous gift in celebrating that history. I also want to acknowledge the other people who make today possible. Kathy Marmon worked really hard. Lisa Wahlberg runs a very tight ship in the department. Without administration, there is nothing. And finally, I want to acknowledge the ASM. You know, the ASM is unusual among organizations in that it actually celebrates and promotes its history. It has a Department of History. So journals that focus on this. They maintain archives. So in the process to get here, Marie had to work with the ASM. And in particular, the ASM then had to vet everything that we were claiming. And it led to some interesting history, reappraisals of history. We may hear some of that tonight at dinner when Karen Thomas will tell us that this was in fact the first school of public health. I want to thank the ASM, which has been represented here by my friend, Virginia Miller. Virginia is an eminent microbiologist, bacteriologist, who is the president of ASM. I want to thank Stefano. He couldn't make it. He's an alumnus of the School of Public Health. And Colleen Putterbaugh, the ASM archivist, who worked to do this. But most importantly, to get here required a village, including a village that is no longer here. It required those discoveries, many others, to build that tradition. And that village had a mayor, and that mayor was Marie. And I want to everyone to acknowledge this and join me in a round of applause to her.
Thank you, Arturo. What has been said here today has been largely MMI-centric, but if you look at the back of the program, you'll see reproduce the text of the plaque here and a list of the particular accomplishments that we have cited to justify it being selected as a milestone site. And what you'll discover is that one of the crowning achievements of the biological sciences and public health was the development of the polio vaccine. And the work on the polio vaccine done here at the School of Public Health was done by a member of the Department of Epidemiology. And so this is one of the strengths of the school and something I think which may not be well appreciated. And so before the presentation or well, shall we say, unveiling of the plaque, Dr. David Gelantano, who's the Armstrong Professor and Chair of Epidemiology, will offer remarks.
Well, I really want to thank you for including epidemiology in this. Arturo and I worked very hard together for almost the last decade, and so it was really a pleasure to be able to come speak with you. And I come to this, I'm finishing my 16th year as chair of EPI in the school, and every fall is for our new graduate seminar series. I lead off with a history of the department, and much of what you're going to see today we talk about, because I really think, as Arturo mentioned, knowing where you've come from will help you know where you're going. I have taught in this venue for, I guess, the last 15 years. Usually it's full of students. And in the beginning, I used to use chalk, and then I went to overheads, and then slides, and then PowerPoint. And today I'm not using anything. So I just want to communicate with you. And I'm going to keep my remarks brief so we can get to the accomplishments that we're celebrating today. But I think there are really three events that have happened over the last century that I want to bring up, that I think really have been important for our understanding of the dynamics of human disease. The first is the development of the Epidemiologic Intelligence Service at the CDC. I think those of us who do disease surveillance, we'd never have been able to do it without the work of the EIS. And I'm going to just mention that came from Hopkins. The second is the well-known discoveries of David Bodian. I'm not going to take any of Marie's thunder away as she talks about that, and Isabel and the crowd. But I must say, I'm wondering, why is the Bodian chair not in epidemiology? This stuff happens. And finally, I just want to briefly mention the HIV epidemic and the response at Johns Hopkins, because I look around and I see some colleagues here that I've been working with for 40 years. And it really was a way that pulled us together in a way we hadn't before. EIS. In 1951, Abe Lilienfeld, the chair of the Department of Chronic Disease, John Hume, the dean at that time, and Phil Sartwell, who was the chair of epidemiology, were asked to come down to the CDC to help them develop a curriculum for surveillance. And what they did was they trained mostly physicians in the practice of shoe leather epidemiology. And that term, shoe leather epidemiology, was first coined by Wade Hampton Frost. And you heard Ellen mention his incredible work on the flu pandemic. And I must say, everyone in the world now knows what epidemiology is because of that last pandemic. So it's nice not having to explain what we do for a living. So they really have become the gold standard, the EIS, for training state and local health departments in disease surveillance. David Bodian, I will skip because Marie's coming up, but he had quite an interesting career. His fellowship ran out and he had to leave Hopkins. The Department of Epi got a grant which brought him back and let him go on to his discoveries. The third component that I just wanted to mention is actually a bit more personal. And in mid-1981, a series of reports about young patients with rare oncologic diagnoses were coming to the fore. And it was occurring among what we would today call men who have sex with men. Then we had all kinds of different terms, some of which are very unacceptable today. But our infectious disease epidemiology group was very small at that point, and we were sort of a backwater science. We studied diarrheal diseases, and it wasn't very exciting. But we had the opportunity to recruit an incredible ID doc and epidemiologist, B. Frank Polk. Polk came to us from the Channing Lab at Harvard, and he was the catalyst for a program of research that has energized the entire university. And it is absolutely remarkable. And Joe and I were back there in those days, and it's really remarkable how much has come out of this. Today, the Hopkins Center for AIDS Research, which is funded by the NIH, is the nation's largest. We have some $110 million this year in federal research funds. And I attribute this success to our predecessors, Wade Hampton Frost to Abe Lilienfeld to Frank Polk. And it's really been an incredible experience to be in this school and to be so influenced by these giants of public health. We owe our CVs to them, I think. So I want to thank you for your attention, and thanks especially to the ASM for commemorating our successes and contributions to science here at Public Health. Thank you.
Thank you, David. So again, have a look at the back of the program, which enumerates the points which support the award of the Milestone site. And the next section of the program is going to be short talks by four individuals who will detail or will provide some of the details that support the points in the plaque. The first of these will be Dr. Joseph Margolek from MMI, who will talk about the firsts, virology and immunology that I've mentioned already. Dr. Marie Hardwick will follow and tell you the story of David Bodian, which has been leaked by Dr. Celentano. We'll have a video from Dr. Jack Levin, who is at the University of San Francisco, who will tell the story of Fred Bang, one of the chairs of the predecessor of MMI called pathobiology, who developed the lymphocyte, limulus amoebocyte lysate assay for endotoxin, which revolutionized a branch of medicine. And then finally, Dr. Griffin will return and talk about the discovery of respiratory syncytial virus by Robert Chanik in the department. So first, Dr. Margolek.
Thank you, Gary, and pleasure to be here on this wonderful occasion. And we've been hearing a lot about the importance of history. And what I want to do is give you a little sense, and only a little sense can be provided, of the man whose vision, persistence, genius, and unbelievable industriousness led to the first, at least some of them, maybe most of them, that we're commemorating today. And you know about the establishment of the school. We've just heard about that. You know about the establishment of this Department of Molecular Microbiology and Immunology, all in 1916. But who was this man, and how did this vision evolve? So I'm going to tell you a little bit about William Henry Welch. Now, he grew up in a small town in northwestern Connecticut, Norfolk, which then is about 1,500 or 2,000 people. He came from a medical background. His father and grandfather were highly respected, even revered physicians in that town who held local, statewide, and even national office. His father was a senator and a congressman. And he had uncles and brothers who were physicians. So he grew up in a small town atmosphere with a tradition of service, as well as a broad perspective. He went to Yale, graduating in 1870, where he was third in his class and elected to the Senior Honorary Society for Campus Leaders called Skull and Bones. I'll come back to that in a minute. I actually wanted to stay at Yale and teach classics, including Greek, but they didn't offer him a faculty position. So he went to a prep school for girls in upstate New York, which he taught at for 1 year, and then the school closed and he was at loose ends. He didn't know what to do. So he fell back on his family for advice, and they advised him to go to medical school. And they had it in mind that he would come and join the family practices in Norfolk, but he developed other ideas. And instead of going to medical school right away, he went to the Sheffield Scientific School, which was part of Yale, and had been established a few years earlier by none other than, among others, Daniel Coit Gilman, who was the first president of Johns Hopkins and whose mission in life was to generate new knowledge. As it turned out, Gilman was still at Yale when Welch was there and had been a member of Skull and Bones also. And it's hard to avoid the speculation that their paths crossed and that might have led to this, because Welch described being in Skull and Bones as the most important thing that happened to him at Yale. After that, he did go to the College of Physicians and Surgeons in New York, and he wanted to do lab work. But the only lab work available was anatomy dissection. So he went into that and did autopsies and earned the trust and respect of the rather obsessive professor who was doing that at Columbia. And it was P&S, it wasn't Columbia then. And then he was advised by faculty there to study in Germany, which was a common thing for physicians to do at that time. And he spent 2 years going to various cities in Germany and learning histology, physiology, experimental pathology, and inflammation. He came back with a firm appreciation of the evolving science of clinical investigation, pathology, and the need for scientific investigation as a profession which hardly existed in the United States. So he tried to set up labs in New York, and it was very difficult. He finally scrounged rooms at Bellevue where they provided space but nothing else. So he had to scrape up microscopes and pathological materials and actually started the first laboratory course at any US medical school in 1879, which was a big hit. It was very popular among the students. It was quickly attended by all medical school students, or students from all medical schools in New York. And he became an authority, even though he had no support for this and had to support himself by other means, which was also common in that time. He was interviewed for the new Johns Hopkins Medical School that was being formed, and the interviewer, who was John Shaw Billings, said he was the best man in this country for the Hopkins, and he was the first faculty member hired at Johns Hopkins Medical School by Daniel Coit Gilman. While he was waiting for the school to be actually built, he went back to Germany and got more training, including in Berlin with Robert Koch. So he finally came to Hopkins in Baltimore in 1885. And that is a picture of him taken after his appointment in 1884. I'm not used to saying 18. Now in 1886, He gave a talk on some of the humane aspects of medicine. This was to the 10th anniversary of the Johns Hopkins University. And I want to just highlight that he's talking about prevention. The great objects of medical teaching include diagnosis, prevention, and treatment. And the prevention of disease is the highest aim of medical science. And then he goes on to talk about how the large medical schools in Europe have developed this in the last few years with hygienic laboratories investigating causation of diseases, the laws governing the origin and spread of epidemics, and principles of various other aspects of public health, all that pertains to the preservation of the health of communities and the warding off of preventable diseases. So clearly, public health was on his mind, and how to do it and the broad scope of a training program that would be needed to advance this in the world. And he ended up writing or co-writing a report proposing an Institute of Hygiene to the General Education Board of the Rockefeller Foundation. This had been a long time in the generation, but I want to quote it because of his definition of hygiene, which was part of the name of the school when it was established. He intended it to include both hygiene and sanitation, the whole body of knowledge and its application relating to the preservation and improvement of the health of individuals and of the community to the prevention of disease. And among the more important subjects he listed were sanitary parasitology, including bacteriology and immunology. So those are the things that led to the departments that you've been hearing about, immunology, which he was the first chair of, although he tried to recruit somebody else before he took the chair, and also the Department of Bacteriology, which he also chaired, in addition to being the dean. Now, the course in virology you've also heard about, initiated by Charles Simon, who came here and also worked without support with great passion to assemble specimens and develop laboratory techniques that were also very popular with the students and led to the Department of Virology being established in 1925, all of which time Welch was still the dean. So I think it's not really possible to encompass the many contributions of this man. I've tried to focus on the department. There were many, many other aspects to what he did, but that gives you the flavor, I hope. Thank you.
Thank you all for being here. And I thank Diane for getting us together to celebrate 100 years of this history. And for all of the rest of you and all your comments, but I only gave myself 5 minutes to tell you about David Bodian and his team. So I'm going to jump into that. Oops. So if you need the slide. But all great science starts from other great science. So I just mentioned a few. So poliovirus was a big health problem in the 1800s. And then, as you heard already, there were many epidemics in the 1900s. And primarily in the summer months throughout much of the world, and estimated in total in the US to affect in one form or another 500,000 people. And I remember at the time that my parents were discussing how scary it was. So nobody knew where it came from, what it was doing, and it seemed to pop up in unexpected places. And now, these are some of the people to remember, and you've already heard the name Wade Hampton Frost several times already. He and Simon Flexner from the Rockefeller didn't always agree with each other on their opinions, but I would say that it was together their debate that stimulated the field to think harder and more critically. And then Jean McNamara in Australia, who at age 15 graduated from the University of Melbourne, very bright young lady, who with her more famous collaborator published in 1931 evidence that immunological differences existed between strains of poliovirus. So very early recognition of this concept that they would be solidified later here at Johns Hopkins. But it's written about her that, excuse me, that she spared no egos in prosecuting the cause. And she recognized the benefit of taking serum from people who had recovered from polio and giving it to those who were ill. And of course, we know that as convalescent plasma today. But I think that was a very forward thought on her part, although she was, that idea was squelched for quite a while, but she dedicated the rest of her life to caring for poliovirus patients. But this left, even those important findings, left many unanswered questions that would take them more than 2 decades longer to get to a vaccine. Like, how was the poliovirus transmitted? And then what was the route of infection of people? And how did it get to the nervous system? And Dorothy Horseman, who was a contemporary of the Bodians at Yale, I think contributed importantly to this scene. She's credited for feeding poliovirus to monkey primates as opposed to other routes of infection to demonstrate transmission. It was a very important observation. And she also did us a service by recording the history in that field. In one of her articles, she lists all of the failed experiments. There was one at the beginning where a monkey contracted poliovirus, and then there were a dozen more papers over several years where nobody could reproduce the finding. So I just want to emphasize the importance of negative data, because there's a good reason why it was negative. We just had to figure it out later. All right, so this was the scene in many hospitals. And we started using iron lungs about 1928 for poliovirus patients. And of course, you heard in the news in March of this year that we lost the last of the poliovirus iron lung residents after 70 years, dying at the age of 76. And then these are the ones to celebrate today. That's David Bodian, Isabel Morgan, and Howard Howe. And Howard was the director of the poliovirus program, which he initiated in 1937 in the School of Medicine. And David Bodian, who had gotten his degrees from the University of Chicago, had become a research fellow in anatomy here in 1939. And Howe attempted to retain Bodian here, as you heard from David, but the funding was not secure enough. And so David took a faculty job elsewhere, only to return soon after, because Howe was successful in landing a nice, healthy funding source for poliovirus research. And at that point, then they moved the poliovirus program from the School of Medicine to the School of Public Health, where then, as you heard, David Bodian was appointed in the Department of Epidemiology, and Howard Howe had an adjunct in epidemiology. And then David Bodian realized, or knew, of course, as you heard some of the history, how important the immunology was. And so he recruited Isabel Morgan from the Rockefeller, who had this expertise, to come and join the team. And this was about 1944. And then for the next few years, the team published many studies leading to answer those questions that I posed a few moments ago, and establishing the key knowledge that was required for Jonas Salk and others to develop those successful vaccines that then were distributed widely. I think the first lot of that vaccine went to like 30 million people in the United States. And it was a joyful time because finally there was a solution. It took more years than it did for COVID, but at any rate, David Bodian's wife, Eleanor, who loved this school dearly and sometimes would escape, if I may say, from her residence. And you would find her here in the hallways for a few years before we lost her. But anyway, her recollection of Jonas Salk was that he always called the house at the most inopportune moments because she was trying to get the 5 kids to bed. And apparently he did call the house. Jonas Salk called many times, as did others, to seek the advice of David Bodian. And in fact, they became very good friends with Isabel Morgan, whom they even made a godmother of one of their children. And so Isabel Morgan, she received her PhD from the University of Pennsylvania in 1938. And as I mentioned, joined in 1944 to share her immunology expertise. So we celebrate here today their ability to build the framework on which our life-saving poliovirus vaccines were made. And for this, they are recognized among several others as the heroes of the poliovirus era. And of course, you have no trouble identifying Isabel, being the only woman recognized here, other than the first lady is standing here. Okay, Eleanor Roosevelt. And next to her, to your right, that is Howard Howe. And then in profile is David Bodian, who's also bumping shoulders there with Jonas Salk. Okay, thank you. The next picture is a picture of the Bodian family. So we can credit them for continuing our legacy. Sorry. This is the Bodian family when they visited us, and they are continuing that legacy.
The next item of the program is a video from Dr. Jack Levin, who's professor at the University of California, San Francisco, and who was a colleague of Fred Bang. Fred Bang was the chair of MMI when it was called pathobiology, and as I've said before, developed the assay which was used and I think is still used for detecting endotoxin. And so if we could cue the video.
My name is Jack Levin. I'm a professor of laboratory medicine and medicine at the University of California School of Medicine in San Francisco. But I was only a research fellow in the hematology division of the School of Medicine when Dr. Bang called my chief, Dr. Connolly, and asked if Dr. Connolly could provide a hematologist to come to Woods Hole and work with him to carry out further studies of blood coagulation in the horseshoe crab. Now, this request was based on a study that Dr. Bang had carried out a decade ago, in which he showed that blood coagulation in Limulus, the horseshoe crab, was triggered by a gram-negative organism. Why he waited a decade to follow up these studies is unclear, but at any rate, I arrived in Woods Hole in the summer of 1966, where Dr. Bang introduced me to the horseshoe crab, about which I previously knew nothing. Dr. Bang didn't believe in providing a lot of detailed guidance to his trainees. He gave you a minimum amount of information and expected you to figure it out on your own. Now, the studies Dr. Bang and I performed, which are shown on this slide, led to the final paper at the bottom. This paper provides the basis for the Limulus amoebocyte lysate test for bacterial endotoxins. When this work was reviewed by the funding agency, we were told that they had no interest in further support because our work had absolutely no clinical significance. Now, if this group of reviewers came back after 60 years, they would have learned that for the past 40 years, the Limulus amoebocyte lysate test for bacterial endotoxins, totally based on the work that Dr. Bang and I performed, has been the gold standard for the detection of bacterial endotoxin in injectable medications, intravenous fluids, and implantable medical devices. And it's currently estimated to be performed 17 million times annually throughout the world. Now, this is a picture of Dr. Bang early in his career, and I estimate this was taken when he was a fellow at the Rockefeller Institute. He subsequently joined the faculty in the School of Public Health and spent the entire rest of his professional career at Hopkins. This is a more formal picture of Dr. Bang. He was a formidable person. He spoke quite quietly, but people paid attention to what he had to say, and for good reason. This is a picture of Dr. Bang in his office of the chairmanship of Department of Pathobiology, which is the department which precedes the current Department of Molecular Microbiology and Immunology. If there was ever a match between the title of the department and the department chair, it's the name Pathobiology for the department that Dr. Bang headed, because he was an extraordinary biomedical investigator with an enormous range of interests, which included hepatitis, parasitic diseases, and pertinently bacterial endotoxin. You get some insight into the range of his interests was that during the period I worked with Dr. Bang, he alternated his summers between the Marine Biological Laboratory, Calcutta, India, where he headed up the diarrheal disease unit, and Roscoff, France, which was the French equivalent of the Marine Biological Laboratory. But my favorite pictures, of course, are those of Dr. Bang and myself. This was taken during a winter course which Dr. Bang organized at the Marine Biological Laboratory. As beautiful as Woods Hole is in the summertime, Woods Hole is dreary and horrible in the wintertime when it snows and is cold, which was the case when we were there. And the last picture shows Dr. Bang and I in front of what was then the new dormitory at the MBL, and I think the smiles on our faces indicate what an enjoyable time we were having. Now, Dr. Bang retired at age 65 and planning to move to Woods Hole with his wife, Betsy, where he would continue his research investigations, which undoubtedly would continue to be productive and where he would have contributed to the scientific community. At the time of his retirement, Dr. Bang was planning to study Sipunculus, a marine worm, which he discovered had a goblet cell which produced mucus. And he thought that by studying mucus production from the goblet cell in Sipunculus, he could gain insight into mucus production in cystic fibrosis, which, as you know, is characterized by abnormal mucus production. Sadly, Dr. Bang never had a chance to continue these studies because he died suddenly while in transit at LaGuardia Airport.
Horseshoe crabs. Okay, the final talk about the points on the plaque will be delivered by Dr. Diane Griffin, who will talk about the discovery of respiratory syncytial virus by Chanik and his associates.
So this is a milestone with the shortest history. That's linked at least to MMI. The first time, Bob Chanik spent most of his career basically at the NIH. But the very first time I met him, he said, I was on the faculty at the School of Public Health, and during that time, we discovered isolated and characterized respiratory syncytial virus. Well, RSV is an incredibly important pathogen. We're still working on getting vaccines for this virus. That caused, at that time, was recognized to cause mainly respiratory, often very serious respiratory illness in young children. So he worked actually with not only people in MMI, but also epidemiology to basically isolate this virus from young children who had this respiratory illness. And back in the laboratory, we had Dr. Roisman, who was at that point, I don't know if he was a postdoc or a student, but he was a trainee in the lab, and who was a experienced virologist, and showed that this was an RNA virus, which was an important concept for this particular pathogen. And they, in addition, worked out the epidemiology of the fact that the virus, once they had it isolated, they could show that it was a very important cause. It was the important cause of respiratory illness in young children. So RSV was from humans was first isolated in the department here at the School of Public Health and characterized as an RNA virus.
It is at this point where we would be preparing for the dramatic unveiling, which we'll do sort of, mentally. We have two representatives from the American Society of Microbiology with us today. Dr. Virginia Miller, who's the president of the ASM, professor of genetics and microbiology and immunology at the University of North Carolina in Chapel Hill, and Colleen Petterbaugh, who is an archivist and a historian, trained as a historian, who, as Dr. Constantine has already indicated, kept us honest in the formulation of the application for the plaque. Stefano Bertuzzi, who is an alumnus of the institution and MPH from 2011 and CEO of the ASM, was unable to attend. So Dr. Miller will deliver a few remarks, and then we'll pretend to unveil the plaque. So.
Well, good afternoon. It is just such a treat to be here. It's one of the real pleasures of being ASM president, of which there are many actually. But I am very honored to be here to share this occasion with you in my capacity as president of the American Society for Microbiology, and to officially designate the Department of Molecular Microbiology and Immunology at Johns Hopkins Bloomberg School of Public Health as a Milestones in Microbiology site. By all standards, as we've heard much of this afternoon, the Bloomberg School of Public Health has met and exceeded the criteria required for recognition as a Milestones of Microbiology. In accordance with its core mission to promote and advance microbial sciences, ASM established the Milestones in Microbiology program to foster greater awareness and appreciation of the microbial sciences as a whole, and to stimulate our interest in our microbiological heritage. The Milestones in Microbiology program aims to promote the microbial sciences and honor significant contributions in the field. These designations highlight achievement that profoundly impacts scientific inquiry and broaden our understanding of microbiology. The Bloomberg School of Public Health, founded in 1916 by bacteriologist and pathologist William Henry Welch, has been a pioneer in public health and microbiology. From the outset, the school housed basic science research laboratories and was the first departments dedicated to immunology and virology. Under Welch's direction, the school soon became the preeminent leader in public health and microbiology. Renamed in 2001 to honor Michael Bloomberg's support and commitment to public health, it became the first independent multidisciplinary school of public health in the United States. The creation of the school followed the growing recognition that an effective public health service was necessary for the prosperity of the country, and that professionally trained public health officers should be essential to this effort. Since the school's inception, the faculty members and their teams and what are now the the W. Harry Feinstone Department of Molecular Microbiology and Immunology, as well as the Department of Epidemiology, have achieved numerous milestones. I will mention just a few of them here, which we've also gone over in many other ways today. First, the creation of the first Department of Immunology in 1916 under William Welch's leadership. Welch's research group had previously isolated the toxigenic pathogens that were responsible for gas gangrene, and this stimulated plans to conduct parallel work in his laboratory. Welch saw that this new school has a chance to pursue this goal. Next, introduction of the first formal course on filterable viruses in 1922 by Charles E. Simon, who had collected viruses not only from across the United States, but also worldwide. And he wrote Welch in 1923 that we are now in possession of what is probably the largest collection of filterable viruses that exist in the world. We also have done here the first delineation of paralytic poliovirus pathogenesis, as was outlined earlier, and identification of the 3 pathogenic serotypes by Isabel Morgan, David Bodian, and Howard Howe between 1941 and 1949. Bodian, Morgan, and Howe worked together to demonstrate that the poliovirus was transmitted via the intestinal tract, that 3 distinct subtypes of the poliovirus are important for disease, and that when combined into fixed or live vaccines could elicit protective antibodies to prevent neurological disease in primates, as well as elicit antibodies in children. We also saw here the first identification of the bacterial component responsible for endotoxin, LPS, induced coagulation test by Frederick B. Bangs in 1953. This led to the development of the Limunus amoebocyte lysate, or LAL test. And this test has been used for decades to rapidly and reliably detect endotoxin contamination in foods, medicines, medical devices, and as you heard, remains the gold standard today after all this time. And lastly, I'd just like to mention, of course, that this here was the first isolation of respiratory syncytial virus, and from humans by Robert Chinook, Bernard Roisman, Ruth Myers, as well as Lawrence Feinberg in 1956. So for more than a century, the research in microbial sciences has flourished in the Department of Molecular Microbiology and Immunology at the Johns Hopkins Bloomberg School of Public Health, producing far-reaching contributions that have advanced the field and generally benefited humanity. The institution's continuing commitment to scientific and educational excellence guarantees many future groundbreaking contributions to further enhance microbial sciences, hopefully by many of you in this room today. In recognition of these achievements and the institution's ongoing leadership in advancing scientific discovery, the American Society for Microbiology is pleased to designate the Johns Hopkins Bloomberg School of Public Health as a Milestones in Microbiology site. So congratulations to the department, to the school, to all of you. It's a much-deserved honor, and it's a pleasure for me to be here today.
Hang on for a second. Okay, this is for the photographer, right? Marie, would you come up, please? So an appropriate reaction would be a gasp of surprise and delight. So each of you will grab a bit of the thing here, and on the count of 3, reveal the plaque. 1, 2, 3. Okay. So, Dr. Casa-Duval, closing remarks?
Just want to thank everyone again. It's been a remarkable, happy day of celebration. We celebrate our past. We hope that 100 years from now, ASM will put another plaque. At least the plaque will be there. We'll have a first plaque. But for now, I ask everyone to join us for a reception where we can have some drink and some light food. Thank you.